Hello, everyone. My name is Mark, and welcome to this new episode of the mitoPodCast. For those of you joining us for the first time, the mitoPodcast is a new program that we hope will build awareness in the mitochondrial medicine, research, and patient advocacy community on topics related to mitochondrial disease and health, as well as to highlight cutting-edge research in the field. Our audio format episodes are approximately 10 minutes in length and will feature the exciting work of an expert in the field in a way that can be understood by all in the community, regardless of educational background. Included with every episode will be an infographic summary of what was discussed, so please check that out as well. Today, I am delighted to be joined by Dr. Walid Houry. Dr. Houry is a professor in the departments of biochemistry and chemistry at the University of Toronto. He also served as acting chair for the University of Toronto's Department of Biochemistry in 2018. Dr. Houry obtained his Master of Science and PhD in chemistry from Cornell University. He then did his postdoctoral training at the Sloan Kettering Institute in New York City and at the Max Planck Institute for Biochemistry in Munich, Germany. Dr. Houry is currently an associate editor of Frontiers in Molecular Biosciences and is on the editorial board of Journal of Biological Chemistry as well as Microbial Cell. Dr. Houry has received many awards throughout his career, including the Visiting Scientist Award from the National Research Foundation of South Africa, as well as the Tokyo Biochemical Research Foundation Award. Dr. Houry, it is my pleasure to welcome you to the mitoPodcast.
Thank you for the opportunity.
Well, thank you for taking the time out of your schedule to join us today. So going to our first question, Dr. Houry, your group is interested in the area of cellular stress responses and the role of molecular chaperones and ATP-dependent proteases in these responses. Could you explain to listeners what role mitochondrial biology plays in some of the pathways that you study?
Yeah, so my group is really interested in this very fundamental question of when a protein is made on the ribosome, how does the protein know what structure it has to fold into, what assemblies it has to be part of, and what activity it has to attain. So to that end, the cell has these quality control mechanisms, or these chaperones and proteases that ensure the proper folding and assembly of these newly made proteins. And these quality control mechanisms exist in the nucleus, in the cytosol, but also are present in the mitochondria. And indeed, so we've been studying a couple of these chaperones and proteases in the mitochondria. One of them that we've been working on for quite a bit is the ClpP, which is one of the major proteases in the mitochondria. And we're trying to understand its basic mechanism of function. But it turns out also that mutations in ClpP lead to many mitochondrial diseases. So one famous one, for example, is Perrault syndrome, which is a disease of hearing loss and ovarian failure. Other sorts of disease are erythropoietic porphyria, which is basically an issue of heme biosynthesis. And also there's ClpP seems to be related to several complex 1 instability diseases. So that's what eventually really led us, is the basic science eventually led us to becoming interested in mitochondrial diseases.
Thank you for that, Dr. Houry. Thank you for walking us through that. So now can you explain some of the most recent findings in your research?
Sure. So in our work on ClpP, we've been trying to understand its mechanism of function. So it's basically a cylindrical type protease. It's made up of 14 subunits that form this cylinder. It's a serine protease, so it has a serous acid catalytic triad, and it has 14 of them, which are inside that cylinder. But what's really important about ClpP is that it works with a cap, which is called a ClpX, for example. It's a cap that really selects what proteins have to be degraded. So it's selectivity filter for the ClpP. And we're trying to understand how this whole complex works. It's a quite complicated system. And what are the targets for this complex in the mitochondria, for example? At the same time, we became also interested in targeting it because, as I mentioned, to this connection. So in targeting it, you can either inhibit the protease. So there are some inhibitors that we identified that block the activity of this protease, but also you can what we call activate it or dysregulate it. So this turned out to be a very neat way, scientifically speaking, to target a new protease. So what happens in the case of the ClpP, what we found is that there are these compounds that would bind to the protease and prevent the cap from binding. So you don't have the cap anymore, but what they do as well is they allow for the entry of larger proteins unspecifically. So they open up the entrance pore, and as a result, you have these proteins that can enter into the ClpP core, and they get degraded unspecifically. So now you have a protease which is activated or dysregulated, chewing up other proteins unspecifically inside the cell, and this would lead to cell death. So it induces apoptosis in many cases. So if it is, to make it maybe clearer to the speakers, basically you have something that's chewing up any protein in your cell, and then the cell cannot handle it anymore. And what happens is that the mitochondria starts the apoptotic pathway. So this was really intriguing. It was an unexpected finding that we could do this for ClpP in the mitochondria. So now we have a mechanism where we can regulate ClpP. So if you want to inhibit it, we can do that. If you want to activate it, we can do that. So we can regulate its activity. And to us, that's really exciting, especially in relations to many of these mitochondrial diseases. Because then in some instances, in some diseases, the ClpP is not as active, so we can try to see if we can push its activity up. In other diseases, maybe it is too active and we can downregulate.
Well, that sounds really exciting. So moving on to our next question, what are some steps you would like to take going forward, and how do you see your work translating into diagnosis and treatment? I think you touched on that a bit earlier in the last question, but yeah.
Yes. As I mentioned, basically most of our work has been concentrated on playing around with the ClpP itself. So the next step is also to see if we can have compounds that might regulate the cap itself, which is called ClpX. It's an ATPase. And you can think of ways where you can maybe inhibit the ATPase activity or induce the ATPase activity. So we would like to go down that route from a basic science on drug development effort to see if we can play with the cap as well. On a broader scale, I'd really like to start collaborating maybe with clinicians who can test some of these compounds in some disease models that they have, whether it's animal models or maybe tissues, that we can then test whether these compounds have any disease curing potential, basically. But at the same time, whether there's issues of side effects and other things that we have to consider when we are trying to do these drug developments. And I should mention really that we've been working very closely with several of the chemistry groups at U of T to develop these compounds, and that collaboration is ongoing.
Great, thank you, Dr. Houry. So, as you know, the development of new treatments for disorders involving mitochondrial dysfunction is not easy. In your view, how can we accelerate the therapeutic development process in a careful and responsible way that prioritizes patient safety? And I guess in a broader sense, is it even possible to achieve this balance of ensuring patient safety while developing drugs quickly?
Yeah, that's a difficult question to answer, or it's a complicated question, basically, right? The problem with mitochondrial dysfunction is that it's really a multi-system disease that often involves the nervous system. And so despite many advances currently in understanding the pathophysiology of these mitochondrial diseases, the clinical management, in many cases, if you look at the literature, if you look at what's happening in the clinic, seems to be largely really supportive. So in many instances, what you are trying to do in the clinic is to play around with the activity of the respiratory chain complexes. So you have things like the myosin or the thiamine that's given to patients, and those try to increase, for example, respiratory chain substrate availability. You have other things like succinate, where you try to bypass a certain respiratory chain. In this case, you're trying to bypass the complex one. So I think for the progression to happen in drug development, you really need to have maybe large-scale randomized trials are, of course, of critical importance. And that might be an issue because maybe in many mitochondrial diseases, you have a smaller cohort, but we need to somehow expand that cohort in these clinical studies. Another thing that's really important in clinical drug discovery is basically to have a valid biomarker or an outcome measurement, right? So if I'm to go back to the ClpP example, For example, I can say that in a normal person, the ClpP activity should be at the level X. And can we find compounds that allow that activity to go back to level X if ClpP is at a level Y? So if we have a clear biomarker for the clinical study, maybe that would help. But to do this drug development in a faster way, that might be more difficult. I think what we need to see happen is that these drug developments have to be done in a more rigorous manner where we have a clear, valid outcome. Now, that mentioned, there's also novel, as you know, there's novel therapeutic approaches that are based on enzyme replacement therapy, for example. So those are coming along, and I think maybe the potential is there for moving forward.
So from the perspective of a research scientist, how do you think a network of patients, researchers, and physicians collaborating together benefits mitochondrial medicine in research? And so in other words, how can patients benefit from those of diverse backgrounds coming together to solve pressing issues in mitochondrial medicine and research?
Yes, I think having really invaluable. An example, a personal example, whereby I was asked by Dr. Anna Andriazza to give a presentation at the MitoNet meeting. And initially I thought it seemed to be maybe too clinical, the MitoNet meeting, because I'm more of a basic scientist, of course. But I wanted to go to see what's going on. So at the meeting, there was this professor from UCL, University College London. Her name is Shamima Rahman. And she gave her talk. She does pediatric metabolic medicine. And she gave her talk, and of course, she described the different patients she was working with and the mutations they had. And it wasn't ClpP related at all. But then I gave my talk on ClpP, and then Professor Rahman came up to me and said, "We are collaborating." And I said, "Why are we collaborating?" So it turned out she had a patient that had a mutation in ClpP itself.
Oh, wow.
So it was just fantastic coincidence. And therefore, now it's the first time I ever encountered someone who has a patient and a protein I'm directly working on. And this started this whole new potential collaboration with Dr. Rahman to see what are the characteristics of that patient, how the ClpP plays a role in the etiology of the disease for that patient. And we hope to continue this collaboration. So this is a personal example of a benefit for such an interaction between the scientist, the clinician, and the patient.
Well, that sounds very exciting, Dr. Houry. So thank you for giving that example. So before we wrap up this episode of the mitoPodcast, do you have a final message for the listeners? And where can they contact you if they would like to learn more about your work?
So I think a support for this combination of a basic scientist, clinician, and the patient is really extremely important. So supporting things like the MitoNet community is very important for the progress, especially in this difficult field of mitochondrial diseases. They are not easy diseases to treat. They are more of a developmental diseases. And having this combination of insights from different angles is really important. And that's something I really appreciate being part of. And anyone who has any questions for me can always reach me at my email address, walid.houry@utoronto.ca. I'm also on Twitter. It's WAH_Canada. And also my lab website is lab.walidhoury.com. Thank you.
Great. Oh, thank you. Thank you so much, Dr. Houry, for coming on the program today and for discussing your really innovative and fascinating work. Before we end this episode, I'd like to extend my thanks to all our listeners. If you have any comments or suggestions for future episodes, we would love to hear from you. Please head on over to our website to download our episodes and to see the upcoming events we have for this year. We have some exciting initiatives planned, so please stay tuned. Thank you all once again, and tune in to our next episode featuring another expert doing exciting work in the field of mitochondrial medicine.